Number 1. The Filchner-Ronne ice shelf sponges. In February of 2021, a team from the British Antarctic Survey lowered a camera through a narrow hole they had melted in the ice. They were not hunting for animals. They were drilling through the Filchner-Ronne ice shelf in the southeastern Weddell Sea to sample the mud on the seafloor far below. The borehole was only about 20 centimeters wide. The ice above it was roughly 900 meters thick, and when the camera reached the bottom, it did not find soft sediment. It struck a solid boulder. On that boulder, something was living. The lead scientist, Hugh Griffiths, published the discovery in the journal Frontiers in Marine Science, and it forced a rethink of where life can hold on. Clinging to the rock was a single stalked sponge and about 15 other sponge-like forms, along with what may have been stalked barnacles. Picture that. These are sessile animals. They cannot move. They anchor to one spot and filter food from the water drifting past them. But there is no sunlight here. This boulder sits 260 kilometers from the nearest open water, under 900 meters of ice, in water hovering near -2 degrees Celsius. Nothing can photosynthesize in that darkness, so no food is made locally. Every scrap of nourishment has to travel horizontally, perhaps 600 kilometers or more, carried on cold currents against the odds. How do filter feeders survive where there is almost nothing to filter? That is the mystery Griffiths could not solve, because the hole was too narrow to collect a specimen. The camera was all they had. Consider the scale. 900 meters of ice overhead is like stacking 3 Eiffel Towers on top of these animals, and because polar life grows so slowly, these colonies could be centuries or even millennia old. The boulder itself likely rafted here frozen inside a glacier, then dropped to the seabed as the ice melted. There are 3 ways to read this. 1. Some faint distant current delivers just enough food. 2. Chemical energy from the seabed feeds them without sunlight. 3. There is a food source down there we have not yet identified. Whatever the answer, it means the ice has been hiding entire communities we never knew existed. And if it hides this, what else is it holding? Number 2. Panagrolaimus Kolymaensis. The last section showed us life preserved in place. This one shows us something stranger. Life that stopped, waited, and then started again. In July of 2023, researchers published a finding in the journal PLOS Genetics that reads like fiction. A microscopic roundworm, pulled from permafrost near the Kolyma River in northeastern Siberia, was thawed in a laboratory. And it woke up, the worm began to move, it began to eat, and then it began to reproduce generation after generation through a process called parthenogenesis that needs no mate. The team, led by Anastasia Shatilovich, dated the surrounding sediment and found the worm had been frozen for somewhere between 45,000 and 47,000 years. That is the late Pleistocene, the same era as the woolly mammoths. To grasp that span, 46,000 years is roughly 9 times the length of all recorded human history. This worm has been in suspended animation since before writing, before farming, before cities existed. Genetic work identified it as a brand new species named Panagrolaimus Kolymaensis. So how does an animal survive tens of thousands of years frozen? The answer is a state called cryptobiosis, in which metabolism slows almost to zero. The worm floods its cells with a sugar called trehalose, which shields its membranes as it freezes and dries out. Theodor Kurzschallia of the Max Planck Institute helped lead the biology, and his team showed the species shares dormancy genes with the common laboratory roundworm Caenorhabditis elegans. The original sample had been dug in 2002 from a fossil rodent burrow 40 meters underground. When skeptics said the worm might be a modern contaminant, the answer was that the dating was done on the ancient sediment layer, not on the worm alone. This shattered the old record for animal dormancy, which had been measured in decades, not millennia. Think about what that implies. If a worm can pause life for 46,000 years, the line between living and dead is far blurrier than we assumed. Cryptobiosis is now a serious avenue for research into preserving human cells and tissue. And there is a colder implication. As permafrost thaws across the Arctic, dormant organisms may be reviving on their own, with no scientists there to watch. A worm is one thing, but what about something built to spread? Number 3, the Guliya glacier viruses. If a frozen animal can return to life, the next question is obvious and unsettling. What about the things that make us sick? In July of 2021, a study in the journal Microbiome, led by Ji-Ping Zheng, cataloged viruses locked inside ancient glacier ice. The ice came from the Guliya ice cap on the Tibetan Plateau, more than 6,700 meters above sea level. Cores were drilled in 2015 by a joint team from Ohio State University and China, with the veteran glaciologist Lonnie Thompson leading the field campaigns. The deepest ice they sampled was about 15,000 years old. That ice formed before farming, before writing, before the first villages. Inside it, the team found 33 groups of viruses. 28 of them were completely new to science. Now, before you imagine a plague, here is the reassuring part. Most of these were bacteriophages, viruses that infect bacteria, not humans. They were tied to bacterial genera like Methylobacterium that also turned up in the ice. To be sure the viruses were genuinely ancient and not modern contamination, the researchers invented an ultra-clean method. They shaved off the outer layer of each core, washed it with ethanol, then rinsed it with sterile water, peeling the ice down to a pristine center. Matthew Sullivan of Ohio State co-authored the ecology analysis. What makes this work matter is what a glacier actually is. It is a time capsule, laying down dust, microbes, and viruses in datable layers year after year, like the rings of a tree. Read those layers and you read the deep history of the planet's climate and its smallest life. But there is a warning folded into the science. Glaciers worldwide are melting fast, and as they retreat, they can release organisms locked away for millennia. There are 3 ways to weigh this. 1, these are harmless phages that will never touch us. 2, some ancient microbe could find a modern host and cause trouble. 3, the viruses themselves are a sideshow, and the real danger is the carbon, the melting freeze. Most ancient microbes almost certainly cannot infect us. But the honest truth is that very few have been studied at all. The Tibetan Plateau is sometimes called the third pole for the sheer volume of frozen water it holds. And a virus is a simple thing. What happens when the thaw exposes something far more complex? Number 4. The Bdelloid Rotifer revived after 24,000 years. A worm came back. A virus can persist. Now raise the stakes again. In June of 2021, a study in the journal Current Biology described a multicellular animal frozen for 24,000 years that revived and then cloned itself. The animal was a Bdelloid Rotifer, recovered from permafrost in the Alizaya River region of northeastern Siberia. Rotifers are microscopic, but they are true animals, with tissues and organs far more complex than any bacterium. The researchers, including Lyuba Shmakova and Stas Malavin at the Soil Cryology Laboratory in Pushchino, thawed the sample and watched the rotifer come back to life. Then it began to reproduce, asexually, producing dozens of genetically identical offspring. There are no males in this group at all. Every Bdelloid is female, and they clone themselves through parthenogenesis. To test how tough the survivors were, the team froze the descendants and thawed them again. They lived. The previous revival record for a rotifer had been about 10 years. This one beat it by more than 2,000-fold. So how do they do it? Like the Siberian worm, Bdelloid rotifers enter cryptobiosis, and during the long freeze they repair the damage that slowly builds up in their DNA. They are already famous for surviving conditions that should kill anything: radiation, complete drying, even the vacuum of space. Malavin made a sharp point about the result. The more complex an organism is, the harder this kind of survival becomes. A bacterium going dormant is impressive. A whole animal doing it for 24,000 years is astonishing. Bdelloids also carry strange genomes stitched together with foreign DNA borrowed from bacteria and fungi, which may be part of their toolkit for endurance. The sample itself was drilled with equipment designed to avoid any modern contamination, and it likely froze fast as the ground locked up near the height of the last ice age. There are 3 angles to hold in mind. 1, the animal simply froze so quickly it never fully died. 2, its real trick is relentless DNA repair. 3, those borrowed foreign genes give it powers ordinary animals lack. Whatever the mechanism, a clone of a creature from the last glacial maximum is dividing in a lab right now. These are tiny survivors. But thaw does not only expose the small. Sometimes it opens the ground itself. Number 5. The Batagaika crater carcasses. Leave the laboratory now and stand at the edge of a wound in the earth. In the Yana River basin of Siberia's Sakha Republic, there is a scar so large and so strange that local people call it the gateway to the underworld. Its real name is the Batagaika crater, though it is not a crater at all. It is a mega slump, a collapse in the ground caused by thawing permafrost. Today it stretches about 1 kilometer long and reaches up to 100 meters deep, and it is still growing, widening by roughly 10 to 30 meters every single year. That is a soccer field's length of new ground opening each year. Its birth traces back to the 1960s when Soviet-era logging stripped away the forest that had insulated the frozen soil. Without that cover, the ground began to thaw and cave in, and it hasn't stopped since. What makes Batagaika a scientific treasure is what its walls expose. The permafrost here dates back an estimated 650,000 years, among the oldest in all of Eurasia. Julian Merton of the University of Sussex has studied its layered walls, which preserve a record of multiple ice ages and warm periods stacked one atop another, and the thaw disgorges bodies. Nearby, researchers have recovered a foal about 4,400 years old, along with ancient bison, and from the wider region, a cave bear and cave lion cubs. The Batagaika foal was so well preserved that scientists drew liquid blood and urine from it. As the ancient ground rots, it also releases long-stored carbon into the air as carbon dioxide and methane, which warms the climate further. The people who live nearby hear it. Each summer brings cracking sounds and fresh collapses. The scar has doubled in size in about 2 decades and is now visible from space. There are 3 ways to read Batagaika. 1. It is a human-made wound triggered by that first deforestation. 2. It is climate-driven, a symptom of a warming Arctic. 3. It is now self-accelerating, feeding on its own thaw in a loop that cannot easily be stopped. Yakut folklore treats the place as cursed, and it is easy to see why. But the bodies it releases carry more than flesh. Sometimes they carry something that can still kill. Number 6. The Yamal zombie anthrax outbreak. For a long time, the danger of frozen pathogens was theoretical. Then, in the summer of 2016, it became real. On the Yamal Peninsula in northwestern Siberia, an unusually brutal heat wave pushed temperatures above 35 degrees Celsius. That heat thawed the active layer of soil deeper than usual, and it uncovered the carcass of a reindeer that had died about 75 years earlier. The reindeer had died of anthrax. Anthrax is caused by a spore-forming bacterium called Bacillus anthracis, and its spores are among the most durable biological structures ever studied. They can lie dormant in frozen soil and bone for decades, even more than a century, and remain able to infect. When this carcass thawed, the spores were released into the soil and water. Grazing reindeer picked them up, and the infection spread through the herds and then to the people who tend them. More than 2,300 reindeer died. Dozens of people were hospitalized, and a 12-year-old boy died. Russian authorities airlifted troops into the region and declared a quarantine zone. What made the event so chilling was the timeline. The last anthrax outbreak in that area had been in 1941, 75 years before. In effect, a disease that had been frozen and forgotten woke up and killed again. Scientists were blunt about what it represented. It was a preview. Across Siberia, old cattle burial grounds hold many carcasses of animals that died of anthrax, and vaccination of reindeer in the region had lapsed in the decades before the outbreak, leaving the herds exposed. There are 3 ways to understand what happened. 1. It was a freak heat wave, a one-off. 2. It was a symptom of systemic permafrost thaw that will keep happening. 3. The deeper cause was human, a lapse in vaccination that left the animals defenseless. All 3 carry truth, and researchers warn that anthrax may not be the worst of it. Other pathogens, even historic ones, could in principle persist in frozen corpses, waiting. The response at Yamal was grim and physical. Infected carcasses were burned and buried deep with disinfectant. A pathogen rides out of the ice on a body. But what happens when the bodies themselves come back whole and far below the ice? Number 7. Subglacial Lake Mercer's buried fauna. Descend now deep beneath Antarctica to a lake no sunlight has ever touched. Lake Mercer lies sealed beneath about 1,067 meters of ice, more than a kilometer of it, in West Antarctica. In late December of 2018, a project called SALSA, short for Subglacial Antarctic Lakes Scientific Access, melted a clean borehole down to the water using a hot water drill. The lead investigator, John Priskew of Montana State University, and his team were reaching into a place that had been cut off from the atmosphere for thousands of years. What they pulled up shocked them. In the sediment cores were the carcasses of tiny animals. Tardigrades, the famously tough microscopic creatures known as water bears, and crustaceans, small shrimp-like forms, and possibly a rotifer. These animals were not alive, but their bodies were there, preserved in the mud, evidence that this dark, buried place had once supported an ecosystem. Their presence suggests that the region was not always sealed in ice. At some point, it may have held ponds, streams, or a coastal habitat where these animals lived. Byron Adams of Brigham Young University analyzed the remains, and chemical dating suggested the animals had lived within the last 10,000 to 120,000 years. That was surprising because Lake Mercer is dark, cold, and starved of nutrients. Mercer is only one of nearly 400 known subglacial lakes hidden under the Antarctic ice sheet, an entire buried world of water. An earlier drilling into nearby Lake Willands in 2013 had found only microbes, so finding whole animal bodies was a real step up in complexity. SALSA used strict sterilization to keep from contaminating the pristine lake. There are 3 ways to read the find. 1, the bodies washed in from ice-free areas like the Transantarctic Mountains. 2, they are the relics of a local ecosystem that once thrived here. 3, they are a preview of what future melting might expose across the continent. This work also feeds directly into the search for life beyond Earth on icy moons like Europa and Enceladus, where oceans hide beneath thick ice. The bodies themselves may have washed into the lake from ice-free areas like the Transantarctic Mountains, carried by ancient meltwater long before the ice sealed the region shut. Whatever their origin, the discovery expanded the idea of where complex animals can persist, or at least be preserved beneath a continent of ice. Preserved bodies under the ice are one thing when they are microscopic. But the next ones come back with fur and faces. Number 8, the Tumat puppies and Ice Age cave lion cubs. Some of what the permafrost gives back is so intact that it stops you cold. Near the Selok River in Siberia's Sakha Republic, prospectors found what became known as the Tumat puppies. The first was recovered in 2011, the second in 2015. And both are roughly 12,000 to 14,000 years old. They are among the best preserved Ice Age canids ever found, with fur, teeth, and internal organs still intact. One puppy's stomach even held the tissue of a woolly rhinoceros, which suggests an adult had brought food back to feed it. Scientists still debate whether these animals are ancient dogs, wolves, or some extinct lineage in between. Then, there are the cave lion cubs Sparta and Boris, found in 2017 and 2018 near the Semyuliak River. Sparta is about 28,000 years old. Boris is around 43,000 years old. Sparta is so extraordinarily preserved that her fur, whiskers, teeth, and internal organs all survived the freeze. Cave lions, the species Pantheris balea, went extinct roughly 12,000 years ago, so these cubs are windows into a predator no living person has ever seen. Both still had their milk teeth, meaning they died when they were only weeks old, likely buried by a sudden landslide or collapse that froze them almost instantly. Love Delane of the Center for Paleogenetics in Stockholm studied the specimens, and the DNA they preserve feeds into serious discussions about de-extinction. CT scans revealed their intact skeletons and soft tissue without a single cut being made. But there is a race here. Yakutian permafrost has become a hotspot precisely because thaw is exposing so many carcasses, and it is ivory prospectors, not scientists, who find most of them, tunneling into the frozen ground with high-pressure water in search of mammoth tusks. Some carcasses degrade within hours of being exposed. There are 3 ways to frame these cubs. 1, the debate over whether the puppies are the ancestors of dogs. 2, the rapid burial that made such flawless preservation possible. 3, the promise and the ethical weight of the DNA they carry. From cubs frozen in the ground, the story now turns to what the sea gives back when the ice pulls away. Number 9. the Larsen Sea newly exposed seabed. In July of 2017, one of the largest icebergs ever recorded broke free from Antarctica. It was called A68, and it calved from the Larsen Sea ice shelf on the Antarctic Peninsula. The scale is hard to hold in your mind. A68 covered about 5,800 square kilometers, roughly the size of the American state of Delaware, and it weighed on the order of 1 trillion tons. When it drifted away, it did something no research vessel could ever do on purpose. It lifted a lid off the seafloor, exposing a stretch of ocean bottom that had been sealed beneath ice for as long as 120,000 years. That is older than the first modern humans to leave Africa. Under an international agreement, such newly exposed areas are designated as special zones for research, and the clock started ticking immediately. The British Antarctic Survey mounted an expedition aboard the research ship James Clark Ross in 2018, but thick sea ice blocked them from reaching the exposed zone. A German expedition on the vessel Polarstern managed to get through and document what was there. They found animals already living on that newly revealed seabed, sea anemones, sponges, sea stars, and other bottom-dwelling creatures. Some of these may have survived under the ice shelf itself for millennia, in near-starvation conditions, feeding on whatever faint trickle of nutrients reached them. Now, suddenly, they were exposed to change. Katrine Lindsay of the British Antarctic Survey helped lead the biological survey, and the goal was urgent: to sample this pristine community before fast-growing species arrived and colonized it, erasing the record of what had been there first. This was not the peninsula's first collapse. Larsen A fell apart in 1995, and Larsen B disintegrated dramatically in 2002, both linked to warming. There are 3 ways to interpret the Larsen Sea seabed. 1. It holds relic communities that were slowly starving under the ice. 2. It is a fresh canvas about to be overrun by rapid colonizers. 3. It may hide chemosynthetic life that feeds on chemicals rather than sunlight. A68 eventually drifted north and broke apart near South Georgia. The shelf gives up sponges and sea stars, but the ground gives up giants. Number 10. The frozen woolly mammoths, Yuka and Lyuba. This is the animal everyone pictures when they imagine the ice giving up its dead. The woolly mammoth, Mammoths primigenius, went largely extinct about 10,000 years ago, though a small population clung on at Wrangel Island until roughly 4,000 years ago. And thanks to thawing permafrost, mammoths keep surfacing. In 2007, a reindeer herder named Yuri Kuti found a 1-month-old female calf on the Yamal Peninsula. She was named Lyuba, and at about 42,000 years old, she is the most complete mammoth ever found. Her preservation was helped by lactic acid-producing microbes that essentially pickled her body, protecting it from decay. In 2010, a juvenile mammoth called Yuka was found near the Oyogosiar coast. Yuka is roughly 39,000 years old, with intact skin, fur, and even preserved brain tissue. And in 2013, a mammoth on Maly-Liakovsky Island produced something almost unbelievable. When researchers cut into it, dark red liquid blood flowed out, still liquid even at below freezing temperatures, hinting at some natural antifreeze property in the animal's blood. Semyon Grigoriev of Northeastern Federal University recovered many of these specimens. The preservation is so good that scientists have pushed at the edge of what is possible. In 2019, a team at Kindai University in Japan took cell nuclei from Yuka and transferred them into mouse egg cells. The nuclei showed brief, limited signs of biological activity. This was not cloning, and no mammoth was revived, but it showed faint embryos still present in cells tens of thousands of years old. Today, the company Colossal Biosciences is pursuing a project to engineer mammoth-like traits into Asian elephants. Behind all of this is a driver that is easy to forget. The reason so many mammoths are appearing is that the permafrost is melting, and illegal ivory trade sends prospectors digging ever deeper into the thawing ground to find tusks. The Wrangel Island mammoths, before they vanished, suffered a genetic decline from inbreeding. There are 3 ways to see these carcasses. 1. Marvels of natural preservation. 2. The raw material for de-extinction science. 3. An ethical question about whether we should revive a lost species into a world that has moved on. Every one of these frozen giants is a data-rich time capsule, decaying the moment it is exposed, and now everything the ice has held is coming due at once. Step back from the individual creatures and ask the deeper question. Why does ice preserve so perfectly? The answer is physics. Cold slows chemical reactions and starves the microbes that drive decay until rot nearly stops. Permafrost is simply ground that has stayed frozen for 2 or more consecutive years, and some Siberian permafrost has held that state for more than 650,000 years. Above it sits the active layer, which thaws each summer and refreezes each winter, while the deeper permafrost normally stays locked. Preservation gets even better when a body is buried fast, by a landslide, a mudflow, or a collapsing riverbank, freezing it before decay can begin, and sealing it away from oxygen in waterlogged, airless ground. At the molecular level, some organisms take active steps to protect themselves. The Siberian worm made trehalose sugar to guard its cell membranes. Certain animals produce antifreeze proteins. And in the case of the mammoth calf Lyuba, lactic acid microbes pickled the tissue, tilting the chemistry toward preservation instead of rot. Glacier ice does something related but distinct. It traps air, dust, microbes, and viruses in datable annual layers. So an ice core can be read like the rings of a tree, decoding both climate and biology going back thousands of years. Underlying the most extreme cases is cryptobiosis, that state where metabolism drops so low it is nearly indistinguishable from death, yet life can resume. This is where a crucial distinction lives. Some preservation gives us a body that is intact but truly dead, like a mammoth or a cave lion cub. Other preservation gives us dormancy, a worm or a rotifer that can actually be revived. The poles and the deep sea excel at both because they combine cold, darkness, and stability like nowhere else on Earth. And cryopreservation labs now try to copy these very mechanisms. There's a spectrum at work, a kind of ladder of preservation. At one end sits a mammoth, intact but unquestionably dead, its cells too damaged to ever restart. In the middle sits specimens like Yuka, dead yet holding faint biological embers that flickered briefly in a laboratory. And at the far end sit the worm and the rotifer, not dead at all, merely paused, ready to resume the moment the ice released them. Understanding where any given specimen falls on that ladder is the whole game, because it decides whether we are studying a corpse or waking a survivor. But preservation is only half the story. The other half is what happens when the ice lets go. The carcasses are the visible part. The invisible cargo may matter more. Permafrost is estimated to store around 1,500 billion tons of carbon, which is roughly twice the amount currently in the atmosphere. When it thaws, microbes begin digesting that ancient organic matter, and they release carbon dioxide and methane as they feed. Methane is especially dangerous because over short timescales it traps far more heat than carbon dioxide. This sets up a feedback loop. Warming thaws the permafrost, the thaw releases gases, and those gases drive more warming, which thaws more permafrost. The Batagaika megaslump is a visible symptom of exactly this process, a self-widening scar releasing stored carbon as it grows. Alongside that carbon come ancient bacteria and viruses, and here, the record splits into the proven and the theoretical. The Yamal anthrax outbreak is the proven case, a frozen pathogen that killed again after thaw. Beyond it, the risk is real but unmeasured. Researchers have revived microbes hundreds of thousands of years old, and frozen human remains from past epidemics have raised concerns about diseases like smallpox persisting in old corpses. The honest position is that most ancient microbes almost certainly cannot infect modern hosts, but very few have ever been studied, so the true risk is simply unknown. Meltwater also acts as a conveyor belt, carrying thawed material into rivers and out toward the oceans. There are 3 ways to weigh all this. 1, the pathogen risk is manageable and often overstated. 2, there could be sleeping giants among these microbes that we would not see coming. 3, the microbes are a distraction, and the carbon is the real threat to the planet. To put the scale in perspective, releasing even a fraction of that stored carbon would rival the emissions of entire industrial nations, which is why the Arctic is sometimes called a carbon time bomb. Monitoring networks now track Arctic thaw in near real time, using satellites and ground sensors precisely because these exposures are not isolated events. They are arriving together now on the same timeline. Every discovery in this video sits inside a race, and it is a race against decay. Once a frozen carcass is exposed and begins to thaw, it can degrade within hours to days. Soft tissue rots, DNA breaks down, and the priceless information inside it drains away. The problem is who finds these specimens first. Across Siberia, it is not scientists, but ivory hunters who uncover most of the carcasses, blasting into the permafrost with high-pressure water hoses in search of mammoth tusks, because a legal ivory trade makes that digging profitable. They are looking for ivory, and the frozen animals are a byproduct. Institutions like the Mammoth Museum in Yakutsk race to preserve whatever finds reach them, but the window on each specimen is short and closing. In the subglacial and under ice sites, the challenge is different. There, teams must drill cleanly to avoid contaminating pristine environments, as Salsa did at Lake Mercer. And on the newly exposed Antarctic seabed at Larsen Sea, scientists had to reach the site before fast-growing colonizers arrived and overwrote the record, which is why international rules protect those freshly uncovered zones. Researchers like Love Delane and Semyon Goryev lead the recovery science, working against both biology and time. There are 3 ways to see this race. 1. A science-first effort to recover and study these treasures properly. 2. The commercial reality that salvage, not science, drives most digging. 3. A more cautious view that some of these things might be better left frozen and undisturbed. Increasingly, local people who make finds feed them into formal science through regional networks, turning chance discoveries into research. What we manage to save now will decide what future generations are even able to study. And the lessons reach much further than Earth. Everything the ice has taught us here on Earth is now guiding the search for life elsewhere. Subglacial lakes like Mercer and the far larger Lake Vostok are the closest analogs we have to the icy moons of the outer solar system. Europa, a moon of Jupiter, hides a global ocean beneath kilometers of ice. Enceladus, a moon of Saturn, vents plumes of water from a sea buried under its frozen shell. The clean drilling methods that Salsa developed to reach Lake Mercer without contaminating it directly inform the planetary protection rules that future missions will need. Every time we find life surviving under a kilometer of Antarctic ice, we widen the definition of where life can exist. The Filchner-Ronne sponges show fixed animals holding on far from any obvious food. Chemosynthesis, the trick of feeding on chemical energy instead of sunlight, is the leading model for how life might survive in a sunless alien ocean. Tardigrades, with their almost absurd resilience, serve as a benchmark for what a living thing can endure, including conditions close to open space. And cryptobiosis, the deep dormancy of the worm and the rotifer, hints at how life might bridge the enormous spans of time involved in traveling between worlds. There are 3 broad positions here. 1. Life is rare, and these are lucky edge cases. 2. Life is hardy and probably common wherever the conditions allow. 3. Life hides wherever liquid water persists, however hostile the surroundings. Lake Vostok itself sits beneath nearly 4 kilometers of Antarctic ice and has been sealed for millions of years, making it perhaps the single best stand-in we have for the buried oceans of another world. Missions like Europa Clipper draw directly on this Antarctic and deep-sea research, right down to how you sterilize a probe so it does not seed an alien ocean with earthly life. Each extreme habitat we uncover on Earth rewrites the odds for finding life beyond it. The ice down here is, in a real sense, a rehearsal for the ice out there. Look back across everything that has surfaced and one thread ties it all together. Every creature in this video shares a single trigger, warming, that is unlocking ancient ice. The stationary sponges under the shelf, the worm and the rotifer that came back to life, the glacier viruses, the anthrax spores, the buried lake fauna, the cubs and the mammoths, the freshly exposed seabed. They are not separate stories, they are 3 faces of one process. Revival, as with the worm and the rotifer that woke and reproduced. Release, as with the viruses and the anthrax that thaw, set loose. And exposure, as with the sponges, the seabed, and the frozen carcasses laid bare. The same cold that preserved all of them for tens of thousands of years is now failing. And what returns ranges across the whole spectrum, from harmless bacteriophages to intact ice age predators to pathogens that can still kill. The scientific windfall and the environmental warning cannot be separated because they are the same event. Permafrost and ice shelves are archives, and we are reading them and erasing them in the same motion. Each thaw is a one-time reveal. Miss it or lose it to rot, and that page of the record is gone for good. The poles and the deep sea remain the least explored and most rapidly changing frontiers on the planet. And here is the unsettling core of it. We are learning the ice's oldest secrets precisely because we are losing the ice that kept them. The vault is open. What climbs out of it next depends on what we choose to do now.